And now, what I want to get into are phospholipids. Phospholipids are a lot like triglycerides. In fact, the only difference is that instead of having 3 fatty acid tails, they just have 2. The third alcohol on that it's got the glycerol backbone. The third alcohol has a phosphate group on it instead of that fatty acid. All right, so to go back a little bit here, right? Fatty acids have 3. We're just taking off that last one and putting a phosphate group on it. And that phosphate group can be protonated or deprotonated depending on what the pH is, the pKa values of those. Typically in physiologic pH, it's going to be deprotonated, like our amino acids and stuff like that. And the most abundant phosphoglycerides, or phospholipids, phosphoglycerides, those names can be used interchangeably. A phospholipid is a lipid that has a phosphate group. It's called phosphoglyceride because it has this glycerol backbone on it. And so these are going to be the predominant ones in us and in other biological life forms. And what makes them phosphoric acid diesters is when we take off a hydrogen, or if it's deprotonated, we tack on an R group. Okay? And that R group can be predominantly these 2 types, ethanolamine and choline. Okay? And those are going to confer pretty different characteristics. So if we have ethanolamine, that's going to give us a phosphoglyceride called cephalin. And cephalin is going to be predominantly a structural phospholipid. So it's going to be what makes up cell membranes, or at least shores up the cell membrane to give it its integrity. Okay. If we put a choline on here, we get things like lecithin, and we can turn it into acetylcholine or acetylcholine. You might have heard of acetylcholine. It's a neurotransmitter. Okay, so it's involved in all kinds of different functions, like our neuromuscular system, our sympathetic nervous system, our parasympathetic nervous system. It's a communication signaling molecule. So memory, attention span, motivation to get off the couch and go for a run or a bike ride or study for an exam coming up, are affected by acetylcholine. A lot of toxins and venoms, poisons, do their work by blocking acetylcholine. So tetanus is called lockjaw sometimes, because if you get really bad tetanus, your muscles just contract like this, and your jaw is locked shut because your muscles are contracting, and the synapses, they use acetylcholine as a neurotransmitter to say, okay, relax. All right, now contract, now relax. I'm using acetylcholine right now to just do this, to move my mouth. And so tetanus is a bacterium, it's a Clostridium tetani bacteria that secretes a compound that blocks the receptor. It's a competitive inhibition, so it blocks acetylcholine by binding to the receptors on the synapses that acetylcholine binds to. And so acetylcholine can't bind, and you don't get the message.
To release the...
Yeah. There's other Clostridium bacteria that do this as well. Clostridium botulinum has a similar mechanism. You might know of the toxin, it's called Botox.
Oh, yeah.
For botulinum toxin. Mm-hmm. And it's the only one that I'm aware of that people purposely inject into their bodies. No one's looking to get a little bit of tetanus, but people will inject it primarily into their face to paralyze the muscles in their face to make them look more youthful. They're also using Botox as a treatment for migraines as well. I'm not sure what the mechanism is, but because it disrupts binding of acetylcholine, I'm guessing that the migraines might be an overactivity of acetylcholine, so maybe that's the mechanism of action, but that's me hand-waving. So bacteria have toxins that disrupt this. Reptiles will have, like venomous snakes will have neurotoxins that, or some toxins from snakes are anticoagulants, so you just kind of bleed out. But then other ones can be neurotoxins, so they kill their prey by again disrupting acetylcholine. Sarin gas, biologic warfare. Syria got in trouble. Gosh, it's been a while now. So much crazy geopolitical shit's happened that you kind of forget about Syria, right? 10 years ago, that was the biggest issue. What was his name? Assad. He got busted using sarin gas on. And so what that does is it disrupts the ability of acetylcholine to bind. You don't get that neurotransmission.
So it kills you. Does it like stop your heart from beating because it's a muscle, or?
It can, yeah. Yeah, I mean, it depends on where it gets to, but yeah. And that's pretty fast acting in that way. So cholinergic, you might have heard the word cholinergic. There are cholinergic receptors. Cholinergic receptors, or anything cholinergic, is something that involves acetylcholine for some.
And then lecithin, it also is also like marketed as a
Supplement. Yeah.
Is that for the acetylcholine?
I don't.
Choline
That's a good question. Lecithin, I don't think is acetylcholine, but it is another one that has choline as the arbor. So it's different in like what makes
it's like transformed into acetylcholine like if needed. But okay.
Yeah, I want to say that I could be wrong. I'm not positive, but I think that lecithin might be different in the fatty acids, but I'm not positive. I'll look that up and let you know. I think lecithin, yeah, the supplement. Let's look up right now. Put you on hold. Why do people take lecithin?
This class has got me more interested in all these like supplements you see. It's pretty interesting. Natural herb stuff, but it makes sense that like pharmaceuticals kind of come from nature, and then you kind of like purify it and refine it and stuff. Totally.
But then there's also like the question of like people like take this thing and we think that it like introduces more of this like beneficial thing.
Yeah. Okay.
Really
So.
know, like, what our bodies do with it, so we're just like, throw much of it.
Like the Indian medicine and stuff, right? It's like for thousands of years we've been using this herb for this and it does this, but it's like what is it actually doing? You know?
Yeah, it's like it isn't.
It's interesting.
So you're right. It is converted into acetylcholine.
Okay.
Oh, okay, cool.
What is that?
But I don't know if that's why they take
The
it.
one that's converted to acetylcholine?
It's a,
So it must
it's,
have
yeah, it's
Oh.
converted into acetylcholine. So I'm not sure if it's taken because for that reason to be a precursor, or if it has some other function. But all right. So phospholipids are predominantly recognized as being components of our cell membranes, right? The phospholipid bilayer. And there's a reason phospholipids make up that membrane rather than just fatty acids or triglycerides. Okay. If we had a fatty acid, it has a bigger molecular head, that polar head, and then the non-polar tail. Right? They all have these polar regions and then non-polar regions, and because we're 70-some% water, the polar regions are going to be on the outside in that watery milieu, and then the non-polar regions are going to fold in. If we had, however, just a fatty acid making these up, then you'd have the polar head here and then a skinny tail. And as you stack them together, we'd have these pores, like these regions that are empty space, essentially. And the whole purpose of a membrane is to be selectively permeable with pores that are, that can open and close. Same thing with triglycerides. With triglycerides, you have, anytime you have a double bond, right, it's going to kink. And so these three tend to sprawl out more and become wider than the polar head, right? And so then you wouldn't be able to stack them nicely together. You'd have these gaps here in the polar head region. Okay, so phospholipids are kind of the Goldilocks, just having the two tails, because that third glycerol is phosphorylated. It makes it, a right shape so that we can stack them together and have this membrane that doesn't let just anything in, right? And of course, in this, you've heard of the fluid mosaic model, right? It's not just a bilayer. In cells, you've got all these proteins and channels in there that receive signals, open up, sodium ion channels, things like that, to let things in and let things out, but it's not promiscuous because of this. If it were this or this, it would be more of a promiscuous membrane. Okay, so we spent a lot of time talking about triglycerides, pretty short times talking about really kind of the next few. The next type of lipid is a steroid. Okay, so whereas phospholipids look very similarly to triglycerides, steroids or sterols have a completely unique structure, right? And as I mentioned on Tuesday, you wouldn't look at this and look at a triglyceride and say, oh, yeah, I can see how those are both lipids, right?
Definitely.
But the one thing that does make sterols is this structure here, okay? This tetracyclic combination of rings. And then what we do to that will differentiate it between something like cholesterol or testosterone or estradiol, and so on and so forth. But they all have this as the backbone, whether it's methylated here or has a hydroxyl group here, that will vary. And in fact, testosterone and estradiol look very, very similar. There's just, I think, one major difference in where a hydroxyl group is, but clearly those have profound, even though the molecule is just barely tweaked, right? It's what gives a male lion a mane and a female lion no mane, right? So, as you folks know by now, small molecular changes make all the difference.
Is this numbering standard too?
Yeah.
Okay.
And did Sean do numbering of bicyclic or more rings, or did you
He
have to get into that at all?
kind of glossed over it, but I don't think we had
Yeah.
to do it for a test or.
Yeah,
Yeah.
we talked about it, especially for like bicycling structures and like how to name them like
Yeah.
a little bit, but not really.
There's a standard way, nucleotides, purines, pyrimidines. There is a way to order them and number them, and yes, this is going to be the way that this is going to be the priority of how it's numbered. So a lot of times this third carbon is what's different, and so that's always going to be in this section. That's always going to be the third. It may not have a double bond, or it may have more than one double bond, right? But having these cyclohex, I won't call it hexane because it could be hexene. These hexagons and then this pentagon here, cyclopentane, is going to be common through all of them.
Okay.
And then this is how they're made. You know, I'm not going to spend a whole lot of time on this mechanism. We're not going to see this on the exam or anything. I'm not going to have you close the ring, but it's synthesized using what's called squalene. And so you can see there's no rings initially, right? But it's kind of set up structurally to have these proton transfers forming an epoxide here, and then we have a nucleophilic attack, and that's successive, right? Several nucleophilic attacks to eventually start closing these rings, and then we have resonance stabilization coming into play, closing another ring, and at some point we get a rearrangement. So this R group here is going to flip up. So you can see cortisone and cortisol, they both have that R group flipping up. And so eventually, again, with these successive rearrangements, we get to that common denominator structure for lipids.
So is squalene like a common compound that's like freely available and gets converted into?
Yeah, I mean, you don't have enough of it that if you were, like for instance, people who suffer from anorexia that have very, very, very low fat percentages will often have hormonal imbalances because they don't have the lipids available to make this cholesterol. So squalene is not something that's necessarily abundant, but that is how. But you can make it. So we usually get cholesterol dietarily, right? In fact, some people have too much cholesterol, but in the absence of that, you can synthesize it, and that's going to be common for all of these biomolecules, right? If you're not getting sugar, I don't know if we talked about it a little bit. There's this process called gluconeogenesis. It's to essentially derive precursors of acetyl-CoA, which is what's used to phosphorylate ADP. If you're not getting sugar, you can get essentially the sugar precursors from other non-carbohydrate sources, so lipids, proteins. In fact, you can convert alanine quite readily into pyruvate. there are these enzymes called aminotransferases. Pyruvate, did we talk about pyruvate?
And
So glucose is converted through a series of steps into pyruvate. You gotta open up the ring, right? Because the glucose molecule is going to be in that ring structure. You open it up, kind of cut it in half. And one of those halves gets modified and tweaked and turned into pyruvate. Pyruvate looks a lot like the amino acid alanine. Instead of having an amino group on that alpha carbon, it has a hydroxyl group.
Okay.
So you can take alanine and just remove the amine group and stick on a hydroxyl group, and now you have pyruvate. And then you can turn pyruvate into acetyl-CoA, and then that goes through the citric acid cycle, and then you get ATP. Right? So the reason I'm mentioning all of this is to say that we have all of these inherent reversible processes, so that when certain things are in abundance, we use those, and then when other things are lacking, we can convert those things to what we need. We can turn fat to protein. I mean, it doesn't happen efficiently, but essentially we can use lipids for protein metabolism. Okay. And so, again, with squalene, yeah, it's not super abundant, but in the absence of getting dietary sterols, there is some squalene available to make this.
Okay. So it's interesting. It can come from dietary cholesterol, but it can also be synthesized from base molecules.
Yeah. And sometimes these will get turned into squalene, right? We'll go, again, biology is, except for aging, if aging was reversible, some people claim they have the secret sauce for reversing aging, but
I've been saying that for a while.
But you can go the opposite direction and make squalene.
Wow.
Cortisone, cortisol. Cortisone, that's a topical. It's used for, it's like antihistamine, used for anti-itching and stuff like that. Yeah. And cortisol is very, very similar. We've taken off essentially that ketone and we've just added an alcohol. Cortisol is, when people say that your cortisol levels are spiking, it's usually induced by stress, and if you're not getting a lot of sleep. And cortisol can lead to weight gain because it stimulates this metabolic process of, okay, we're stressed, and we've evolved that when we're stressed, we need to conserve our energy because we need to stay alive, right? And stress back in the day wasn't, I've got an exam coming up, or I have a deadline coming up. It's like, I've got this warring tribe, or I've got a food shortage of some sort, right? And so, different functions with just a very slight change in the functional group. All right. So, next one. Any questions on that? Any other questions? Okay. So, moving a little bit quickly through this stuff just because this tends to be a little bit more, this is more obviously biochem related, so I'm trying to keep it relevant to OChem, and that's kind of those are the more basic OChem things. If we want to talk about it more, we dip into biochem. Prostaglandins aren't, really a better category would be to call these eicosanoids, which I think is on later on the slide. Prostaglandin is a type of eicosanoid, which is a type of lipid. Okay? Prostaglandin is a very common icosanoid, and very biologically relevant. And there are all different types of prostaglandins. So depending on the functional group, right, whether we have ketones here, or in this case we have an alcohol, ketones down here. No ketone, we have just alcohols, peroxides, lots of different flavors of prostaglandins. And a prostaglandin is what is going to signal inflammation in our body. Okay, so if you get injured, you're going to have a release of prostaglandins. Prostaglandins are then going to trigger the inflammatory process. So cytokines, chemokines, immune cells will start coming to the site of the injury, especially if it's been opened up, right? If you bang yourself and there's no like trauma to the skin, you're not going to get an infection, but you will still have immune cells there because they might need to gobble up dead and damaged tissue in turn. And so with prostaglandins, you can take something like ibuprofen. What is ibuprofen? It's NSAID. What does NSAID stand for? Non-steroidal, right? So prostaglandins aren't steroids. Prostaglandins are eicosanoids. And I'll get into NSAIDs in a minute, but also if you have a fever, particularly if you have kids with fevers, you give them ibuprofen. Now, there's a philosophical argument to not, because fevers are there for a reason, to kill off the infected agent. But if you want them to go to daycare, they have to be fever-free for 24 hours. You pump them full of ibuprofen and you say, okay, get to school. What that does is that decreases the amount of prostaglandins in the body. So whether it's a fever or a cut, or you're just sore because you ran a bunch, right, and your body's aching, the aching is inflammation. And the inflammation is from an upregulation of prostaglandin. So you take NSAIDs to decrease the prostaglandin to then decrease the subsequent inflammation.
What is the alpha and beta refer to in these little descriptions?
So whether or not it's pointing up or down. So with sugars, did we talk about alpha and beta with sugars, right? So if the OH group for sugar is pointing up, it's beta. If it's pointing down, it's alpha. So this refers to the different locations of these. So for instance, beta is going to be hydroxy, so that's the hydroxyl group. This is going to be gamma unsaturated, meaning there are positions that don't have a double bond. Beta does have a double bond. So it's just telling us, because these are so different from each other, the alpha and beta tell us a location. They're like a locant. And I think that probably is carried over from biochem. Because OChem, IUPAC, it would just give a number.
So it looks like alpha is like one removed from the carbonyl, beta is two, or
Yeah, from the first carbon. So with amino acids, the alpha carbon, that's where that amine group is. That's going to be one removed from the carbonyl. All right, so this is how we synthesize them. And so prostaglandin is actually made from something called arachidonic acid. And arachidonic acid is an eicosanoid, so it's a lipid. And so the distinguishing feature of eicosanoids, and I think I mentioned this at the very beginning on Tuesday when we looked at the different structures of the different types of lipids, it has this chain that wraps around like this. Okay. So you see it in the prostaglandin. The only difference here is that we've turned it into a ring. And in this case, this is that epoxide version P, prostaglandin P2. But what you need to initiate this is an enzyme called cyclooxygenase. And that's what NSAIDs control to prevent prostaglandins. So it's not like the NSAIDs prevent activation or prevent activity of prostaglandin. What they do is they prevent the arachidonic acid from going through these steps and turning into prostaglandin by deactivating the cyclooxygenase, this enzyme that is responsible for this hydrogen abstraction step. So we remove this hydrogen, and then that leads to this cascade of free radical, right, with the one barb, right? So we have an electron going here, electron going there, and then that allows us to tack on this oxygen bond. And this called is referred to as a coupling. Did you guys talk? Have you heard of coupling before? Okay, so we're talking about bromine radicalization, maybe. Yeah. Okay. So you have this coupling that carries out, and then these come together here, an electron up top, electron on the bottom. It's all just a series of single electrons to eventually close the ring here and give us our final product. And it'll be slightly tweaked depending on what's going to be produced, right? Not all of them are going to have that epoxide. That's just the example for that one. We've got A, B, C, D, E, and F as well. Right? It's going to be a slightly different mechanism, but that's the general process that happens. Okay.
So those substituents, do they get added on like after, at the end of the mechanism, or is that like a different?
No, so they're gonna be added on, I believe, at this step.
Okay.
Okay, so instead of being added on and then turned into the epoxide, I think they break off in this case, because you'll notice that in some cases they have 2 oxygens, some they just have 1, but and that can happen because the O2, you break it off, but we have all these other radicals that can then satisfy that other oxygen. Okay. So depending on where the radicals are going, you'll notice that in this case, all the ones that just have 1 oxygen have a double bond, and it has 2 oxygens, or this epoxide, there's no double bond.
So it's the conditions of what's going on.
Yeah. So we must be taking hydrogens off to form OH, right? And then maybe we're forming water. Yeah. We take off 2 hydrogens, mix it with that other oxygen, and just get water. Here, we're using both those oxygens. So, good question.
So is this all happening while attached to the cyclooxygenase? Until it gets to that final step and then there's further modifications?
Yeah.
Okay.
And so it's not just prostaglandin that comes from arachidonic acid. Arachidonic acid is the precursor for lots of different eicosanoids. Prostaglandin is just being one of those. Okay. And so the arachidonic acid is kind of the common denominator. It's going to be the precursor. So when we take our NSAIDs, we're not going downstream or too far upstream. We're inactivating that enzyme. So arachidonic acid just stays as arachidonic acid. And as you might guess, if you're taking NSAIDs, especially if you're taking a lot of them, or not a lot of them, but chronically, you're taking them regularly, it's not just necessarily operating on anti-inflammation, it's also disrupting other eicosanoid production that have different functions. Right? And so it's not really a great thing to take NSAIDs long term, right? Because it can disrupt other biological processes. Mix it up with some Tylenol or something.
How does acetaminophen work?
Don't know.
It's not an NSAID or whatever.
No, I believe aspirin's an NSAID
Okay.
and ibuprofen, but let's see.
Some roommates from Australia, and they were always like, "Do you have any paracetamol?" I'm like, "What the hell are you talking about?" And then, like months later, I realized they were just asking for Tylenol. I was like, "What do you call it there?" I actually had it the whole time.
Oh, definitely.
Like, no, not paracetamol.
Oh, no, it must be an NSAID. It says it blocks prostaglandin synthesis. I didn't think it was an NSAID though.
Different mechanism.
Well, so I know acetaminophen.
It inhibits COX-1 and 2. So it doesn't, but that's cyclooxygenase, so interesting. So it's the same mechanism of action.
Well, doesn't acetaminophen also like thin your blood, where it's ibuprofen?
Aspirin.
Aspirin. Oh, yeah.
aspirin. That's what it is.
Aspirin does,
Okay.
yeah.
Right, acetaminophen's the alternate.
Because they take it for a heart attack or something.
Maybe aspirin is the non-steroidal and acetaminophen is the NSAID. Acetaminophen. Yeah. Yes, I mean, I should know that because I give my kids Tylenol as well to get their fever down. And if they have a really bad fever, you give them both at the same time.
Yeah, I just remember when I got my wilderness first responder this past summer, like we had a chapter on like pain management, and he was saying to like trade off between the two of them
Yeah,
every couple hours.
but their mechanism of action's the same. It might just be because the byproduct. So once you metabolize those drugs, they have different side effects, right? So taking too much Tylenol can be toxic to your liver. I don't think ibuprofen has the same issue. Ibuprofen can be hard on your stomach though. It can lead to ulcers and stuff like that. So maybe that's why. It's not because the mechanism. It's not because clearly it's not because you're targeting a different mechanism of action, but it might be because of the downstream side effects of using one continually. All right, let's take a break, and then we'll get into terpenes. Terpenes